Hi, my name is Shirley Liu. It is my great honor to be elected for the 2020 Benjamin Franklin Award for Open Access in the Life Sciences. I grew up in China, attended Peking University, where I majored in biology. Then I transferred to Smith College and had the opportunity to double major in biology and computer science. During my PhD training at Stanford, I started doing bioinformatics research, and I also received a PhD minor in computer science. Since 2002, I have been at Boston, and now I'm a professor of statistics, biostatistics, and computational biology in the Department of Data Science at the Dana-Farber Cancer Institute and Harvard University. Over the years, we use the following research approach. When there is a new genomic technique developed, we try to develop the algorithm to help biologists adopt these new genomic techniques. We also collaborate with experimental biologists who are using these techniques to make sense and generate hypotheses from their high-throughput genomic data. We help these techniques to become widely adopted by the community, and when there are many such data generated, we try to integrate large-scale data from these techniques and use tumor profiles, genetic and drug screens to make biological discoveries ourselves. And then we will build integrated computational infrastructure and resources on these public profiles in order to help experimental biologists or clinicians to make discoveries using this public data. Since there are many new experimental techniques that continue to be developed, we do this in an iterative process. And when the next technique was developed, we again start with algorithm development, experimental collaboration, and when there are more data accumulated, we start to make discoveries from data integration, and we help build infrastructure to help others also utilizing these public data. The type of public data we try to model includes hundreds of mouse tumor profiles, thousands of CRISPR screens, thousands of cancer cell line profiles with genetic and drug screens, thousands of tumor profiles with immunotherapy treatment, tens of thousands of tumor profiles not on immunotherapy, millions of single-cell profiles on tumors, hundreds of millions of immune receptor repertoire sequences, and hundreds of terabytes of genomic sequences. Over the years, we have developed many open source computational resources to help cancer gene regulation. These include MAX, for ChIP-seq or chromatin accessibility profile p-calling. Systrome, which is a database of transcription factor histomark ChIP-seq or chromatin accessibility profiles in human mouse, and we also added many related search functions. We also developed LISA to predict transcriptional regulators from differential expression. Maestro for integrative analysis of single-cell RNA-seq and single-cell ATAC-seq data analysis. We also developed open access computational resources to help model cancer immunology and therapy response. They include Timer for estimating tumor immune infiltration from tumor bulk RNA-seq data, TIDE to predict response to cancer immunotherapy, TRUST to infer tumor T-cell receptor and B-cell receptor repertoires from bulk or single-cell RNA-seq data, MAGIC for quality control, analysis, and visualization of CRISPR screens, and CARE to predict regulator genes of targeted therapy response and resistance. Through all these efforts, we follow Benjamin Franklin's advice to do well by doing good. Through developing these open access resources, we were able to identify or interact with many experimental or clinical collaborators. And through this collaboration, we were able to derive very exciting biological insights. Through the generous support from our funding agencies, we were able to recruit more talents to join our experimental research effort, engage the research community, and receive the happy surprise of awards such as the Benjamin Franklin Award. In recent years, there has been some concerning policies regarding international collaborations. And there are a number of papers and books published regarding this. We want to really emphasize that disease has no boundaries. Scientific research has no boundaries. And really open access in life sciences should have no national boundaries either. I want to thank members of the Liu Lab over the years for developing these open access resources for the community. I want to thank them especially for their continued efforts in maintaining and updating these tools and answering questions from the users and engaging in the communities for many, many years after these computational resources are published. I also want to thank my mentors, my collaborators, the funding agencies, and the community. Thank you for your support, and together we can continue to develop computational resources for research and hopefully with the goal of enhancing human health in the long run. Thank you.